talk about the pathogenesis. Okay, how does the disease develop? Well, we talked about the pathogenic process previously. Remember, there's 4 steps to this. We said 4 steps to being a better pathogen. We gave that like a Tony Robbins lecture previously. So our first step of pathogenicity, passage from one host to another. If an organism is going to be a successful pathogen, if they're going to get into the medical books, as if pathogens who want that, they have to infect more than just one host. They have to be able to spread to hosts. They have to be able to, as I said before, they have to be able to franchise and spread out. So the first step, pathogenicity, passage from one host to another. The way Staph aureus does this, or methicillin-resistant Staph aureus, is survival on the skin. Of course, we've talked about the skin. Not a very good environment for most microbes because skin has a lot of, that's right, a lot of salt. And so to survive on the skin, an organism must be a halophile, must be able to tolerate high salt concentrations. And so Staph aureus can survive very well on the skin, particularly for the community-associated or acquired form of MRSA. If it survives on the skin, it can be easily spread from one person to the next. And we tested for this in the laboratory. Remember MSA? Remember what MSA stands for? Mannitol salt powder. It looked like this. Remember, our potential Staph aureus organisms will grow on here and turn yellow. We're not concerned about yellow so much. The mannitol fermentation is not really indicative of the MRSA, but the ability of the bacteria to grow in a high salt concentration helps it survive on the skin. Survival on the skin allows it to be passed more easily to the next host. Also, Staph aureus, MRSA included, can survive well on inanimate objects. Band-aids, towels, something that makes contact with skin can't pass this to the next person. By the way, what do we call an inanimate object that transmits disease? Remember that? Because it's an F? Rhymes with termite? Okay, so a fomite. Okay, and so it's gram-positive, survives well on inanimate objects, and can also be spread that way. So both of these help this to spread from one host to another by allowing the bacteria to survive on the skin or on inanimate objects. Second step of pathogenicity, our organism must be able to get into the host. So it gets to the host and it must enter the host. Okay, so we talked about portals of entry previously, ways that the microbe can get into the human body. The most common portal of entry for methicillin-resistant Staph aureus is the parenteral route, okay, through some sort of break in the skin. Okay, so it rides in on some sort of trauma. Microbes can't penetrate intact skin. MRSA can't penetrate intact skin, but they can get through breaks in the skin. So cuts and abrasions create an opening in the skin to allow microbes to get through. Here we've got some road rash right here, and so as a result of this opening in the skin, methicillin-resistant Staph aureus from the skin could get into this. It could be an autoinfection, a person's own normal flora causing an infection. They can come into contact with somebody else, maybe that was kind of muscular, it could be arm wrestling and pick up the disease from somebody else. Or, once again, an inanimate object or fomite could allow these microbes to get into and cause infection, in this case, and disease. Now the parenteral route, once again, this is more for the hospital-associated. There's a condition called surgical site infections. And as the name implies, this is typically nosocomial. It could be normal flora. It could be the person being infected with their own flora during a surgical procedure, or it could be passed from one of the hospital personnel as well. So you see the initials SSI, that stands for surgical site infections. But both of these are parenteral routes, so the microbe entering the body through breaks in the skin. Now the infection can also affect the mucous membranes. Typically it doesn't enter the body, but we'll go ahead and talk about the mucous membranes. Mucous membrane is the respiratory route. Remember, this is how most microbes enter the body. And in the case of methicillin-resistant Staph aureus, this can cause a necrotizing pneumonia. So we've seen the term pneumonia previously. We know that's the lungs filling up with fluid. What does the term necrotizing mean? So we see necrosis. Necrosis indicates death or destruction. So in this case, it's a pneumonia that causes tissue death or tissue destruction. A little bit like when we talk about the difference between diarrhea and dysentery. You know, diarrhea would be kind of like pneumonia, fluid accumulation. Dysentery with tissue destruction would be kind of synonymous to a necrotizing pneumonia in this case. we've talked about Staph epidermidis causing urinary tract infections. It's not unheard of for MRSA to also cause urinary tract infections. In this case, typically it's going to be nosocomial, not so much autoinfection like we talked about with E. coli, but nosocomial typically associated with catheterization. But by far, the most common way that MRSA enters the body is through the parenteral route, through breaks in the skin. So it survives on the skin, survives on animate objects, it gets through breaks in the skin. Then our third step of pathogenicity is overcoming host defenses. Once a microbe has survived in the environment and it enters the host, then it needs to overcome the host defenses because the host is going to try to get rid of that microbe. And we've spent an entire lecture talking about host immune responses to infections. And so there's a few different virulence factors that Staph aureus has, MRSA as well has, that can contribute to pathogenicity that involves overcoming the host defenses. One is toxin production. These toxins will kill host cells. And one of the most well-known virulence factors for MRSA is this Anton-Valentine leukocidin, or PVL. And we already know what leukocidins are. Remember what kind of cells leukocidins kill? The name itself is a big hint. Leuko for white, it kills. So this is the type of toxin that kills white blood cells. Okay, so as these phagocytic cells, shown here, try to eat up the Staph aureus, these toxins kill off the white blood cells and overcome this branch or this part of the host immune defense. This is a very well-studied virulence factor, thought to be very prevalent, particularly in necrotizing pneumonia, but more recent studies have shown that it doesn't seem to increase the virulence. So it plays a role in protecting the microbe against host defenses, but it may not play a very significant role when it comes to actually causing disease symptoms. By the way, just on the side, we talked about electron, or just gave it away. We talked about microscope shots. What kind of a shot is this? Okay, electron microscope shot, scanning or transmission. Okay, so this is a scanning shot because we're looking at the three-dimensional structures. And once it's been colorized, and we see the cocci here and the phagocytic cell there. So the leukocytes kill off white blood cells like this. Another one, overcoming the host defenses, is being able to attach. We know the host tries to rinse microbes away through mucus or saliva or tears or urine. And so the microbe has a way of attaching. This will aid in the pathogenesis. And it's thought that the methicillin-resistant Staph aureus produces various types of adhesions. And these adhesions will allow the microbe to attach to host tissues. So these adhesions, like the idea of adhesive tape, allows the microbe to stick to something different. Rather than being cohesive, it's adhesive. And these may also contribute to the virulence or the ability of the microbe to cause disease. So we've got toxin production, attachment to the host. Now I want you to add one more. I didn't put on the PowerPoint because I figured that you might remember this one. So I want to add one other virulence factor. Just put this off to the side on the notes. Remember we talked about a particular virulence factor unique to Staph aureus. We tested for this in the laboratory. Remember we said that if it grows yellow on mannitol salt, that's a good indication that it might be Staph aureus. But there's one ultimate test or one defining test. Remember what that was? Starts with a C. It wasn't catalase. That's how we differentiate Staph from Strep. Okay, that's right. It was coagulase. Okay, so remember, like most enzymes, the name tells us what it does. Coagulase causes coagulation. And so remember, this allows the Staph aureus to produce what I call a pseudo capsule. Kind of a false capsule. By coagulating plasma around it, it creates this slippery coating that prevents phagocytosis. So when we talk about